Welcome. This Career Essentials video for PhDs and postdocs is on lay summaries. The video covers what a lay summary is and gives advice on how to write about your research for members of the public. It gives reasons why it's important to be able to write a good lay summary. And then it shows you a framework for a lay summary and examples of how to write in plain English using analogies. The NIHR definition of a lay summary is a brief summary of a research project or a research proposal that has been written for members of the public, rather than researchers or professionals. It should be written in plain English, avoid the use of jargon, and explain any technical terms that have to be included. A lay summary may seem just a small part of your grant application, but being able to write a good lay summary is actually an important skill, and it is worth investing in learning how to do it. That's because it's an opportunity to make a good impression on your reviewers. The lay summary of a research proposal is often the first thing they will read. Also, patients or carers of patients could be part of the funding panel, and the funder may use your lay summary for publicity purposes. Being able to write a good lay summary is essential in other situations too. For example, in patient information and ethical approval for clinical studies. When you are establishing a collaboration with industry partners or scientists from a different discipline. When you are influencing policy. When you are getting funding from the government, it helps them to justify public spending. Or being able to write in plain English will come in handy when you want to write a press release to promote your research. How to write a lay summary? Well, the title should be simple and relevant. Then you have to tell a story about a whole project. You can follow this framework. Discuss the background, the aims, the expected results or the results, and the impact of the results. Cover these questions. Who will be doing the work? What will you be doing? Where will you do it? When will you do it? Why will you do it? How will you do it? And always keep asking the question, so what? Why is this exciting? Why is it relevant? What are the benefits? Why is it timely? The most important thing is to write in plain English. This may sound simple, but actually it's a skill that does not come naturally to everyone. And it's especially difficult when you have a maximum word count. Use simple words and sentences. Many terms used in academic English are either overcomplicated or contain no useful information, such as significantly, virtually, or literally. Avoid jargon. Explain any technical terms that are essential to tell your story. Analogies are a good way to explain complex ideas, even if it means you lose some of the technical detail. Often, the summary is not lay enough. How can you check this? Well, read it out loud to yourself and ask friends or relatives to read it, and then get them to ask you questions. This will help you understand which parts need clarification. Here is an example of a first draft for a lay summary. The maximum word count for this example, which was from a grant report, was 350 words. No title was required. The human body consists of a tremendously large number of around 37 trillion cells, the smallest functional units that work in concert to ensure our bodily existence in the world. Robert Hooke was the first to discover individual cells in 1665, looking down a microscope at a piece of cork. However, it is only now that we are in the exciting position of being able to characterize the identity of individual cells across entire organs, owing to the revolutionary advancements in single-cell genomics. Large-scale initiatives like the Human Cell Atlas, HCA, have the aim to catalogue all cells of the human body, as we will only understand the whole if we understand its parts, and are beginning a new era in our understanding of human health and disease. During my PhD, I contributed to this technological advancement. The workhorse of the HCA is RNA sequencing of single cells derived from digested tissues. RNA molecules tell a cell what functional proteins it needs to produce in order to fulfill its unique properties. Sequencing of these RNA molecules allows a fingerprint of a cell's identity to be generated at high throughput. However, because tissues are digested into pieces, the spatial information of where these cells are positioned in the body is lost. I helped to develop a spatial transcriptomics technique that allows us to map back these cell identities into tissue space at high position and across large tissue areas. Ultimately, the technique developed will contribute to the efforts of the scientific community to complete a full circle of taking apart the human body in its cellular building blocks and reconstructing full spatial maps of its three-dimensional construction. I used the technique to characterize the identity of a particular cell type in the brain, so-called astrocytes, and found that different classes can be distinguished based on their location within the cerebral cortex, the evolutionary most recent part of the brain. These classes may be differentially affected in neurological conditions. In addition, I characterized the RNA profile of the gene X, which is mutated in a neurodevelopmental disorder throughout brain development. The study provides important information for gene and protein replacement therapies that aim to reinstate X protein function in cells that have lost it. This draft follows the framework and explains some of the science in simple language. However, you can still find technical terms and jargon in this version that may make it difficult to read for a lay person. You can now pause the video and go back to try and identify the jargon and technical terms. I highlighted some of the more difficult language, for example, the term single-cell genomics. Can you think of a way to rewrite this without having to use that technical term? Do you think a lay person will know what RNA molecules and RNA sequencing are? Even the term proteins may not be understood by a lay person unless it is put in context or explained. Can you come up with simple ways of describing high throughput or spatial information? And what about spatial transcriptomics technique? Will a lay person know what a cerebral cortex is? Do they have an understanding of evolution? RNA is used again here. Even words like gene and mutated may not be understood by a lay person. What is a neurodevelopmental disorder? What do you think gene and protein replacement therapies and protein function mean to someone who has no or a very basic understanding of the terms gene or protein? Here is the final version of this lay summary. If a title was required, it could be, for example, creating a 3D model of cells in the brain, a step towards treatment for children born with a brain disability. Here's the final version of the text. The human body consists of around 37 trillion cells, small functional units that make up tissues and organs. We can imagine the human body as a large Lego object with bricks representing these individual cells. Just like a Lego castle with all its colored bricks, we can only understand how such a complex object is built if we break it down into its individual building blocks. Recent technological advances allow scientists to digest tissues and read the blueprint for each cell in the human body. This blueprint for the cell's shape and function is called the transcriptome. The next step is to take this catalogue of blueprints for all the cells of the human body and put it back together to uncover the secret of how our bodies are built. But how can we put the pieces of information together if we have digested the tissue and do not know where a cell came from? I contributed to the development of a method to look at parts of the blueprint for a cell in intact tissue by giving it different colors that show up on a microscope. This allows me to visualize the unique identity of cells within their tissue surroundings. By then matching this part of the blueprint to the whole blueprint for every cell from the digested tissue, a whole 3D model of the tissue before digestion can be reconstructed. I used this method to study astrocytes, star-shaped cells in the brain that support nerve cells. Scientists previously thought that all astrocytes are the same. However, By looking at the individual cells, I found that they have different blueprints and therefore probably take on different functions. So by looking at the individual Lego bricks, I found that the brain is more complex than previously thought. In some disabilities, like X deficiency disorder, some building bricks are missing parts when a baby's brain develops. I'm now using my method to find out where exactly the bricks are missing parts, so that we can treat children suffering from this disorder by reintroducing the parts in the right place. Now, can you look at the final version and identify the background, aims and results, and impact? The first paragraph gives the background. Just pause the video if you want to read it again. The second paragraph and the beginning of the third paragraph give the aims and the results. The aim is given as a question. This study aims to find out how we can put the pieces of information together if we have digested a tissue and do not know where a cell came from. The end of the summary gives the impact. If we find out what parts of the bricks are missing, then we can treat children with a disability, X deficiency disorder. Next, I will highlight some of the analogies and places where technical terms are explained. If you would like to try and identify them yourself first, please just replay this part of the video to identify them. Lego bricks are used as an analogy for cells. Individual blocks can have a different shape and color, but together they build a castle. Cells with different shapes and functions make up the human body. A blueprint for the cell's shape and function is used to explain transcriptome. The analogy used here is that by identifying where cells are located in the tissue, by lighting up a part of the blueprint, I can then match this part of the blueprint to the whole blueprint. And that means I know where a cell with that particular blueprint will be located. Perhaps not representative of the exact technical detail and perhaps still difficult to visualize, but definitely language that a layperson can understand. By looking at single-cell genomics, individual Lego bricks, it became clear that there is more than one class of astrocyte. The brain is more complex than previously thought. A cell is missing a protein. A Lego brick is missing a part. Reintroducing parts in the right place is the analogy for gene and protein replacement therapy. Now you are ready to start writing your own lay summary. Please feel free to book an appointment with one of our careers consultants if you would like some tailored feedback on an application, including your lay summary.